Estamos ao vivo.
Boa tarde a todos, conferencistas, palestrantes, equipe Labart e aos participantes que nos acompanham nesse último dia do Sinergia, Ciência, Educação, Arte, via Google Meet, com transmissão aberta pelo YouTube. Lembramos ao público participante que se inscreveu que é imprescindível realizar o preenchimento do formulário de confirmação de presença aqui no chat do YouTube. Seguimos hoje, terceiro dia da programação do Sinergia, uma pequena alteração aqui da programação que havia sido anunciada anteriormente. Começaremos à tarde com a palestra da professora Romina Nassini, da Itália, com a fala Receptor Channels and their Crucial Role in Cancer Pain. Na sequência, então, o professor Bernard Andrieu, com Le Corps Vivant et Sinergia avec le Monde, une esthétique cosmique. Na sequência, então, né, teremos a professora Sônia Alexandra Leite Velho Mendo Barroso, de Portugal, com a fala Lenteptide, an Emerging Class of Drugs. Na sequência, professora Mariela Xereg, dos Estados Unidos, com a fala La Investigación Artística en el Cruce Arte, Ciência, Tecnologia. E logo após a conferência de encerramento com o professor Henning Ulrich, com a fala Purinergic Signing and Neurodegeneration. Passo agora, então, a palavra à professora Romina.
Can I start? Yeah,
yes,
can I start?
Please.
Okay.
You have up to 30 minutes, and
yes.
we kindly ask you to start your speech with your professional presentation, please.
Yes. Okay. Did you see? You see presentation? Okay. Did you see my presentation?
Yes,
perfect.
Full screen.
Yes.
Perfect.
Can I start?
Yes. Please.
Oh, thank you. First of all, I would like to thank the organizing committee for inviting me to this very interesting meeting. And today I would like to talk and to discuss with you some data that we obtained regarding the receptor channels and their involvement in different mechanisms. And in particular, today we will talk about cancer pain. We know that the pain is a devastating condition that affects more than 50% of patients undergoing the treatment for cancer. And more importantly, more than 60% of patients who have advanced metastatic cancer experience pain. Thus, pain is a very untreatable condition. But we asked in our lab, which cellular mechanisms are involved in this condition. We know that the peripheral sensitization and the signal propagation in nociceptor are the important condition for the development, the maintenance of the painful condition. In particular, ion channel and G protein couple receptor that are expressed in sensory neurons have the ability to encode the intensity of noxious stimuli and lead to the transduction of pain signals. Among the different receptor and ion channel, we focus our attention in a specific family of channels, the transient receptor potential channels. They are a big family of ion channels. In particular, I want to remember the vanilloid one, that is the channels activated by the painful sensation of red peppers, or the A1 that is activated, for example, by the wasabi sauce when you eat Japanese food. And we will talk about these channels that could be involved in the developing the maintenance of the painful sensation during cancer growth. I already told you that many factors can be involved in pain induced by the growth of cancer. And I reported here some papers that reported the involvement, for example, of the nerve growth factor. In this paper, we observed that mice injected with the intraplantar oral cancer cell developed mechanical allodynia that is reversed by an anti-NGF administration. And more importantly, the treatment with the anti-NGF also decrease the TRPA1 and TRPV1 expression, but the mechanism is unknown. In another model of cancer pain published in 2017 in a prostatic adenocarcinoma model, we observed, the authors observed the development of the nociceptive response that was reduced by the treatment with the TRPA1 selective antagonist, the first antagonist that was developed. And in particular, they observed the development of cold hypersensitivity. That is another kind of pain developed by the cancer patient together with the mechanical hypersensitivity. Thus, we wanted to explore the mechanism underlying the development of cancer pain. And firstly, we observed in a model of cancer pain induced by the injection of melanoma cells directly in the hinge, the mechanism involved in such mechanism. And we observed that during the tumor growth, there is an accumulation of macrophages inside the tumor. Thus, I mean that the tumor growth in the intraplantar site, the plantar site, and we observe an increase in macrophages population. But we also observe an increase in macrophages also in the sciatic nerve. Thus, we asked if these macrophages were important for the development of pain, because in these mice we observed the induction of the mechanical hypersensitivity. Thus, we used a specific colony of mice that are called the mafia mice, the macrophages phase-induced apoptosis mice, in which with the treatment with a specific depleting agent, we were able to completely deplete the macrophages population. And what we observe? We observe a depletion of macrophages inside the tumor mass, but also inside the sciatic nerve. And these mice recovery from the allodynia. Thus, these mice did not, in these mice, the allodynia was reduced after the treatment. Thus, macrophages were important. But which is the mediator that promotes the macrophages expansion? Among the different mediators that we observed in the sciatic nerve, we point our attention in the macrophages colony stimulated factor. And we observe an increase of this cytokine in sciatic nerve and in tumor. Thus, we asked whether this macrophages colony stimulating factor was important, but in particular, we asked whether which cells is important in the macrophages colony stimulating factor release. We previously observed the cells that were important in the development of pain, and we observed that the Schwann cells are the main actor in different model of pain. Thus, we asked whether MCSF was expressed and was released from Schwann cell. Firstly, we observed that MCSF is expressed in the messenger of MCSF is expressed in Schwann cell, in particular in human Schwann cell and in mouse Schwann cell. And then we tested whether the stimulation of Schwann cell were able to induce MCSF release. And what we observed? We observed that following the stimulation of in vitro stimulation of Schwann cell with the H2O2, they were able to induce the macrophages colony stimulating factor release. Thus, Schwann cell were important in this release. And we asked whether also whether TRPA1 was important in this mechanism. And we observed that if we blocked by the selective antagonist the TRPA1, we were able to reduce the MCSF release. Thus, Schwann cells are important in this release, and TRPA1 is also important. Thus, we tested a MCSF receptor antagonist, this compound PLX, and we observed if the mice were treated with this compound, they were protected from the development of mechanical allodynia as well as from neuroinflammation. Thus, MCSF is important. But more importantly, if to test whether TRPA1 was important for the neuroinflammation and pain, we used the specific colony of mice with the selected deletion of TRPA1 only in Schwann cell, these PLPA1 CRE mice. And what we observed? We observed that if we injected melanoma cell in these mice did not develop mechanical allodynia as well as neurogenic inflammation, neuroinflammation. Neuroinflammation as in these mice, the macrophages in sciatic nerve were absent. Thus, Schwann cell TRPA1 drives the neuroinflammation and pain in this model of cancer pain. Later, in 2023, we wanted to study also another model because as I already told you, the development of pain is more important in people with metastatic cancer. Thus, we developed a model of metastatic bone cancer pain by inducing the growth of breast cells in the breast. So we observed that the previous work described a role of different cytokine in bone cancer pain. For example, here I reported the involvement of IL-17A in bone cancer pain, in a model of bone cancer pain in rats, and also the involvement of TRPA1 and TRPV1, as well as TRPV4. Thus, TRP channels are also involved in this model of pain. Another model, another paper, recent paper, described the involvement of the acid-sensing nociceptor TRPV1 in a breast cancer progression in bone. But no one better described the mechanism underlying this pain, the development of pain. Thus, we observed that the inoculation of breast carcinoma cell in the 40 mammary gland induce the primary tumor, the increase of primary tumor size in the mammary gland, and the development of mechanical lodynia called hypersensitivity. This is the development of mechanical and cold hypersensitivity during the days. We observed we recorded this pain during 20 days. And we also observed the development of non-evoked pain, as well as the induction at day 20 of a metastasis in the bone. Thus, the model were done, but we also observed that the pain-like behavior were associated with the increase of an osteolysis marker, EGF1. In fact, we observed an increase in EGF1 also in the bone and in the sciatic nerve. Thus, we asked whether this cytokine was involved, this biomarker was involved in the development of pain. And first, we used a receptor inhibitor, this PPP, and we observed that the treatment with this inhibitor was able to reduce the mechanical lodynia, the cold hypersensitivity, and non-evoked pain. But it was not able to reduce the tumor, the primary tumor mass, and the metastasis in the bone. Thus, it was EGF1 is involved in the pain development, but not in the tumor growth. Thus, here we used another approach, because also in this case we wonder whether the Schwann cell were important. First, we observed that EGF1 receptor is expressed in Schwann cell, and thus, if to test whether this receptor expressed in Schwann cell, given the importance of Schwann cell in the development and maintenance of pain, was important, we used another approach. We used an adenoviral vector-based thread-dependent EGF1 receptor silencing by using an shRNA for the selective silencing of this receptor in Schwann cell. And what we observed? We observed a reduction of mechanical lodynia called hypersensitivity and non-evoked pain, but not again a reduction in the bone metastasis as well as in cancer growth. EGF1 receptor is also present in primary sensory neurons, but what we observed, if we silenced the receptor, EGF1 receptor, also in sensory neurons by using the same approach, we didn't observe a reduction of mechanical lodynia and cold hypersensitivity and non-evoked pain. Thus, the EGF1 receptor in sensory neurons was not important. But which are the mechanisms activated, the intracellular pathway activated by EGF1 receptor activation? We observed the phosphorylation of different proteins that culminate in the eNOS activation and the production of nitric oxide. And we performed a different in vitro experiment in which we tested the abolition of the response, the nitric oxide production during after the blockade of this receptor. And in vivo, we observed that if we block the intracellular pathway by using a different antagonist, here the NT157 or AKT inhibitor, or L-name to block the nitric oxide synthase, or by blocking the nitric oxide, we observe a reduction in mechanical hypersensitivity called hypersensitivity and non-evoked pain. Thus, this is the intracellular pathway. Intracellular pathway that culminate in TRPA1 activation. Thus, again, if we use the mice with the selected deletion of TRPA1 in Schwann cell, we observe again a reduction in mechanical hypersensitivity called hypersensitivity and non-evoked pain. More importantly, EGF1 in Schwann cell activate TRPA1 to expand again endoneural macrophages. In fact, if also in this model we use mafia mice, also in this case we observe a reduction in the mechanical hypersensitivity called hypersensitivity and non-evoked pain. Thus, also in this case, we observed that Schwann cell EGF1 receptor trigger an intracellular signaling cascade that terminate with the nitric oxide dependent TRPA1 activation and macrophages expansion. Thus, Schwann cell are important to mediate again the painful condition seen in cancer. Thus, in conclusion, the take-home message is the concept that the tumor microenvironment is a big scenario of neuroinflammation and chronic pain. Neuroinflammation that induces the peripheral and maybe the central sensitization. We are studying also the central desensitization. And in summary, we can observe that the study of nerve microenvironment sure could offer targets for novel anti-cancer drugs that target different pathway, TRPA1, as well as the different receptor and cytokine involved in this condition. I want to thank my laboratory based on the Department of Health Science at the University of Florence, our supporting agency, and you for your kindly attention. Thanks again for inviting me.
Thank you, Professora Romina, for your excellent speech and for your collaboration in the CAPES PrInt project for all these years. Thank you very much.
Thank you.
Muito bem, agora então passamos a palavra à professora Raquel para poder apresentar o nosso próximo palestrante.
Bonsoir Bernard, Andrieux.
Bonsoir Raquel.
Merci d'être parmi nous et aussi de ton accueil lors de ma visite CAPES PrInt. Tu as jusqu'à 40-60 minutes de parole, à commencer par te présenter sous l'angle professionnel.
Thank you very much, Raquel. I am very happy to speak with you. Sorry, I am a just philosopher, and French philosopher is not very specific. I work about body and philosophy of body, and I speech my presentation about a new conception that we developed with Raquel and other person in Brazil, but also with my student and my colleague about body ecology, because I think the work in philosophy is more concrete today, because we have many problems about the change of climate and post-colonialism, and we have the possibility to describe a new relation with nature, with other. And I hope to make this description in my communication. Okay. It's okay, Raquel? Yeah.
Ok. O Raul precisa passar o slide. Sim.
Oh, sorry, it's a translation in Portuguese, no in Brazilian language. Sorry, but and perhaps it's better for you. And I present about Sinergia. Sinergia, the problem of connection between corpo vivo and the interconnection in the holistic understanding of the world. And I published with my colleague Teresina Petrushata Nobrega a great book in Liber's Art about emergir, na naturaleza, about body ecology. And I published in English in the Routledge collective handbook about this problem. Next. What is body ecology? Body ecology, it's a new philosophy of body and a reflection about practice and our responsibility about the nature and other. And the problem is not only of change the practice in the world, but also change the practice in the use of my body. And the problem of conciencia corporal está muy importante para la comprensión de la understanding of how we can change my behavior and my relation in the use of my body with other and in the nature. It's a sort of critics of capitalism, and also, and we have the possibility to make a new ethics of existencia, not return in nature. I publish in Portuguese. I discovered this book because I don't know this publication. A book about Nova Filosofia do Corpo. And in this work, we describe 3 point of view of body. One, it's a corpo vivo, the relation of body ecology of my body, and I can make ecologization and modification of my matter. And the second is the construction of habito, by a technique of body, like Marcel Mauss describes this process, or Pierre Bourdieu. And Margaret Mead describes this, and in Samoa population. And third, it's a corpo consciente. Corpo consciente, it's the moment when I have a good perception of my situation. Next. I published 2 other books about my personal history and by my father and my mother, and we make translation in Brazilian language, and we have published this in Natal. It's very important to understand what is my cosmos, where is my relation with my country, and with the relation of my origin. And in these 2 books, it's like Nietzsche say about the history of our body. It's an idiosyncratic perspective. Next. Well, what is the difference between prácticas cósmicas and ecologia corporal? And I propose here a reflection between 3 conceptions of ecology. One, it's ecology, cosmic ecology. It's the consciousness of our place in the world, and the relation with earth, with air, with element, and what is the possibility for in my culture to understand this. Second, it's ecology sensorial, and like Raquel work about this, I think his work is very important because she has in aesthesiology, in aesthetic perspective, the possibility to make a good contact with nature, but his photographic work. And the third, it's ecologia corporal. It's a practice, body practice like yoga, tai chi, meditation, and other technique of consciencia corporal. It's a new possibility to have educação, somática, aesthesiologica, or immersiologia. Next. I think it's not new, my problem about ecology of cosmic. Like you know in Brazil, and about the autochthone culture and traditional ritual, we have many possibilities to describe, as Descola described in his book about nature. And we have many populations to develop ritual, practice, collective practice, and understanding with our relations with nature. And I think it's the age of autochthone culture. It's very important for understanding the space of ecology. Next. Well, what is cosmosis? Cosmosis is a word that I create this word for describe the relation of Sinergia with the cosmos. And I think it's very important, like me in Sao Paulo, this photography describes my contemplation of this tree in the park of the University of Sao Paulo. And the tree is very beautiful in this park. And cosmosis, it's the possibility to have a special disposition of sensation, like Raquel described in this photography and in the work. And I think we can feel this special osmosis with nature. And it's very good for build a new relation with other species, but with other people. And we can respect the integrity of nature. Next. Well, we have a word about this. You speak about Sinergia, but perhaps in French we have in language the term of symbiose. And symbiose is like a Sinergia. Symbiose is sort of the mix of the relation between organism, animal, plant or microorganism, and especially we can describe this synergy between the different elements in a new philosophy of life. Next. Well, if I put just one synthesis about my first point of view. We can describe the emergence, emergence in corpo vivido, that the activity of living body, and like naturismo, cosmos, ecology, and we can feel by practicas corporal the feeling of nature by immersion, emergencia, emergence of this information produced in my living body, and I can feel by empathy or by attention or by feeling this information in my perception. I don't know, it's clear. Raquel, it's okay? Yes.
It's okay.
Okay, next. The effect of this Sinergia or osmosis, it's a vitalidad, vitality, the augmentation of vitality in my body, in my living body. We can have a very good feeling, a new sensation of internal sensation in my body, and we can make new information produce involuntary in my body and emerge in my by despertar or awareness in my conscience. And I think it's a possibility to have a new knowledge of my relation with the world. Next. Well, it's not new. My idea is not new. It's not original. I am a poor philosopher, sorry, because Bachelard and the great philosopher, French Bachelard, and work about this, about the reflection in 4 elements. He has developed a reflection about the imagination of these 4 elements. And I think for the thema of Sinergia, Bachelard, and just I rewriting the development of Bachelard philosophy, but also I want to describe how each element can produce effect into the body. Next. Well, this possibility of vivacity, vivacidad, and as un effect de la auto-organisation del consciousness pour la body ecology. When you are in immersion in element like agua or aire or sun, we can alive immersion and very good feeling or bad feeling, because it's not always good. And but we can feel the life. We can feel the contact with nature. Next. Well, my philosophical project is it's a new synthesis in schematization. And we are here on the Imersao et despertar, es el niveau de body ecology, and conciencia corporal et vitalidad es el niveau de la conciencia de awareness. And we have the possibility to communicate by the immersion of the experience of Imersao or despertar into my vitalidad or conciencia corporal. I think it's a very good project for education or for pedagogy, aesthetic pedagogy, because it's not abstract. It's a very material and sensorial experience. Next. Well, you can develop the body ecology, and we can discover self-health, auto-saudade by this new relation, and you can practice a new experience of body by a somatician, by a doctor, or by also therapist or other practitioner of body. And we describe this like immersiologia. Immersiologia it's a word that we create for translate the problem of emergence of living body in my lived body. Next. Oh, essaye. Dis-moi, Raquel, pour le temps, si je déborde. Hein?
Non, tu peux aller, tu peux finir là.
Next. Well, I can make a difference with body ecology. Body ecology explores the direct relationship of the living body and its nervous system with their environment, especially during immersion in natural, artificial, or virtual environment. The activation of the body is the activation of the nervous and sensitive part of the living body under the effect of ecologization. And for immersology, we propose to analyze the immersion of the living body in the consciousness of the living body. Immersion is the involuntary movement in our body and of network, mood, of image, about which our consciousness knows only the emergent part. Next. Well, we have make a good synthesis about this in two papers. One paper this year about immersology in sport science, and we describe how the unconscious living body can produce a new corporal relation, and also in this book. Next. Well, it's more complicated, sorry, but just for say to you that it's more complex to the passage to living body here on the back, and on the top describing body. We have different steps of emergence, and here we have the possibility to use objective techniques, and here you can use a phenomenological or subjective or artistic technique. And for each level, you can make a communication between objective technique and subjective technique. Next. Well, for the theme of Sinergia, I think that you can read and read again the book of Lynn Margulis. It's very important. It's a book about the microcosmos and Gaia. And we have here a very good interest about symbiosis and porosity of body. And Donna Haraway or Bruno Latour say that it's very good expression because it's the development of phylogenesis, but also in ontogenesis and also in epigenesis. And it's a very good interest for the description of holistic synergy between the all living being. Next. Well, you understand my project. My project is to develop a microcosmos and like an experiences of each person with the relation with the body of the world. And in the better event, we have cosmosis, Sinergia, but also you have the risk like Chernobyl, Fukushima, or I have you live in Santa Maria, a very effect of a catastrophe in the last month. We live this change of climate. And the effect is not always good. The nature can produce a very bad effect. And like this term is dysmosis. Dysmosis is a word contrary of cosmosis. Next. Well, Levi-Strauss describes this in Brazil. It's not new, my perspective of microcosmos. Because Levi-Strauss describe this relation into the autochthone culture. I think it's very important to return at this perspective. Next. Next. Well, we published a very great book in 10 years, and the title of the book is A Body Cosmos, the body of the world. And we publish for each culture, for each autochthone culture, we publish the relation of body of the world, the relation of Sinergia with nature, the relation with mythology, and I think it's very important to develop an aesthetic anthropology and for know today, not yesterday, not in the past, but today, we have the possibility to know the new relation of cosmos. And we prepare with Raquel, we prepare a great book about this question, about cosmos. Next. Well, the consequences is the intercorporeity between the living person. I published this about my last book about the contact with living, but we have a German philosopher, very important, Andreas Weber. I don't know if you know this book, but he's very important researcher. And this perspective is very important because he say, okay, we have intercorporeity in the Sinergia with a living body, but this intercorporeity poses a problem of community. What is a community with other species? What is the possibility to share with the other species a relation of vitality, for example? I think it's very important philosopher actually in Europe. Thank you. Next. Well, it's not new. My work is not new. Okay, and because Raquel Carson, you know Raquel Carson, Silent Sprint, she died by the contamination of DTT. She described in his book the bad effect of chemical work in country, and I propose this organization. Perhaps the Sinergia, it's the problem of common health. And we have two dimensions, dimension of cosmos and dimension of transfrontier. And for cosmos, we have the possibility to have a contact with other living being, but also we can develop the holistic care. Like I say, holistic care is a very good mean for discover the Sinergia and between the relation and on the world, but also we have other side, the transfrontier, and transfrontier is an obligation to rediscover the autochthone culture for understanding the description of cosmos and ecology. I don't know, it's clear, Raquel? Yeah.
Okay.
Next. Wait, and today we have a perspective of indigenizing anthropology. It's a new perspective. It's a perspective to describe by anthropology how the possibility to describe the organization in each culture, this relation of cosmos. Next. Yes, here are some example. Also in the north of continent, Paul-Émile Victor or Nastasia Martin, and describe the life also in the north of the Earth. Next. Well, it's not new. You are in Brazil, a very great anthropologue, Eduardo Viverio de Castro. And it's very important. Also, my colleague in Sao Paulo, Anna Cristina Zimmerman, and his colleague. And we have developed a relation about this relation between cosmos, health, and knowledge. And Eduardo Viveros de Castro critiques Lévi-Strauss because he describes an indigenous conceptual conception and not a structuralist conception. We say it's very important for understanding Sinergia not to have an universal point of view, but a relative point of view. Next. Well, it's not original. Judith Butler described this possibility to describe body that matter of gender trouble she described how we can describe by the deconstruction of stereotype and the norm for discover how we produce the norm by the activity of body. And I think the Sinergia is a very important perspective to describe the process of production of norm. Next. Well, I see this story about Brazil, about the flower of beauty, and it's a very good example of the debate between the past mythology of culture and the today culture, and how we have the problem on transmission of this synergia with nature by the destruction of the forest, Amazonia, or destruction of the condition of life, and the difficulty to maintain and transform the synergia with nature. Next. Well, you know this map. It's a map about the problem of os bem comum de Amazonia, and we have the problem of globalization and the use neoliberal of capitalism, of exploitation of forests, but also we have the possibility to understand how in the local tradition we have the possibility to have a conflict between different of conception of relation of cosmos. Next. Well, Yanomani perhaps or the terra protegida. It's a conception of the protection of Earth and against the destruction of Amazonia, but also against all the destruction of population, and with culture and autochthon conception of synergia with the Amazonia. Next. Well, this is the end of my communication. I have reflection about a different relation with corpo and community, and because in first we can say that okay, for Sinergia we have the possibility to have common body with other species, but the problem is compartilhar. They share this conception with different concepts. Non, c'est bon, c'est pour le vaccin. Ah, pardon. Different conception of my sorry, it's my son, and the third conception, it's the conception of autochthon culture, and the concept of localism, not globality, but the concept of the declaration of locality by the protection and the respect of a difference of culture. Next. Well, we have now a movement about Sinergia, and the movement is we are indigenous, and we have the possibility to describe the health of autochthon community, and revendication by this group about to connect by and respect this connection with the nature and culture and traditional ritual. Next. Well, I developed with my colleague a solution. Perhaps one solution is sport sustainable, slow sport. It's we developed with my colleague about the research of a new use of body in nature and the concept of sustainability. It's a good concept, and it's a new reflection about harness, about a deep ecology. And the conception is a new mode of movement in space and time, like a Nordic walking, mindfulness practice, or ethno-eco-sport exploration. Next. And the last of the last is my experience with Petruccia da Nóbrega. We published this in Portuguese, in Brazil, and in English. It's my discover of naked surf in Praia de Tambada in Brazil. I don't know if you know this place. It's very interesting. We live 5 days in this beach, and it's very interesting the practice of naked surfing and the possibility to have contact with a new body ethics, leisure, and modesty in this relation. Next. Like this, and make a photography on the posada. And my last picture, it's a sort of symbol. Next. And this, we make this photography, and it's very interesting event. It's a very old woman with disability come on the beach with other person and naked, and we help this person to sit and on the beach for make a Sinergia with the sea, with the air. It's a very beautiful image for me of the Sinergia. Thank you very much. I don't know, it's my time. It's okay. Raquel?
OK. Merci Bernard de ta présentation. On est là très content de ta participation.
Merci. Merci de votre invitation. Merci beaucoup.
Merci beaucoup.
À bientôt.
À bientôt.
Ciao.
Ciao.
Ok, então, obrigado, professor Andrew e Raquel. Daremos continuidade, então, com a professora Sônia. Já também peço desculpas pela inversão que nós fizemos aqui nos horários, em função também de outros compromissos dos palestrantes, mas daremos continuidade, então. Bem-vinda, professora Sônia. Você tem até 30 minutos para a sua fala. Limited Tide, Emerging Class of Drugs, e pedimos, por gentileza, que a senhora comece fazendo a sua apresentação profissional ao público.
Yes. Vou começar em português. Queria agradecer à coordenação do projeto CAPES PrInt, em particular à professora Maria Rosa Quitolina pelo seu convite para participar neste evento, e agradecer a presença de todos. So now I will move to English as I was requested. So I come from the University of Aveiro in Portugal, which is near Porto, that probably most of you will know. That is near, well, in the central north Portugal. In my lab, we have 3 research lines. One of them, which is the one I'm going to talk today, is about the search for new antimicrobials, namely, or in particular, antibacterials, which is now the focus of the work I will present, although these compounds might have other applications and other effects, but I'm not going to talk about that today. So, antipeptides is an emerging class of drugs. As you know, antibiotic resistance is a problem worldwide, and there is no way that is going to go back or return in the next few years. I think it's going to get even worse. And also resistance to cancer drugs and other drugs is also a problem worldwide. So the need for new drugs is urgent, and unfortunately, and if we focus only in the antimicrobials, the pharmaceutical industry is not really too much interested in investing in this area because it's not profitable. But other areas are, of course, of more interest, namely cancer, heart diseases, Alzheimer's, and so on. So the problem regarding antibiotic resistance and antibiotics is to really understand or know if the antibiotic pipeline is running dry or not, because we know that the resistance in bacteria is increasing, and new antibiotics development is falling down, and no new drugs are in the pipeline, or only a few, and most of them are used in the clinical context. But regarding these drugs, we know that synthetic drugs can be developed or designed, but nature was, is, and still will be in the future the source of natural products. And these products may be extracted from plants, animals. You probably have heard about antimicrobial peptides, microbial resources, and so on. So nature is still the major source of these compounds, and I'm going to focus on microbial resources. So if we look at the natural compounds of microbial origin, we have lots of compounds with lots of applications like surfactants, antimicrobials, anti-cancer, anti-tumor, cholesterol lowering, pigments, immunosuppressants, and so on. And these compounds are usually belong to one of these four classes, namely terpenes, polysaccharides, hydrophores, and peptides. The peptides is the class I'm going to talk a bit today. And within the peptides, we can have two different types of peptides. The ribosomal peptides that are synthesized from DNA, and the peptide is synthesized as a long molecule that undergoes post-translation modifications and releases the final compound, which is functional. I'm sorry.
Bruno, estou numa conferência.
I'm sorry. In the RIPs, in the ribosomal peptides, I know that you probably heard about nisin, which is a compound that is used in the food industry. And if we look to the non-ribosomal peptides, they are synthesized by multi-enzyme complexes that are arranged in modules and in domains that activate the amino acid and promote the condensation, and the final peptide is then released. One of these compounds is vancomycin, which you probably know, and penicillin, and this is most of the traditional drugs belong to this class. But RIPS is a class of peptides that is increasing in terms of families that have been that are being discovered, especially or mainly because of the new technological advances, namely sequencing of genomes and so on. So more and more compounds are being discovered, and these compounds have some characteristics, like they are typically very small, between 20 and 100 amino acids. They are very low in mass, and they are encoded by structural genes. And here you have the most important families of RIPS peptides, and lantipeptides is the one I'm going to talk about today. So lantipeptides are really peptides that contain lanthionine and methyllanthionine, which are introduced by these post-translation modifications that these peptides undergo to be active or functional. Regarding the bioactivity, they can be used as antibacterial, and they can be used in the food industry, anti-allogenic, antiviral, morphogenetics or surfactants, antifungals, or antibacterials, or other compounds. And many compounds are being discovered with many activities that belong to this class of lantipeptides. The ones that have antimicrobial activity for simplicity, are named as lantibiotics. And if you look at this graph, you can see that the number of lantipeptides that have been discovered is increasing, and the number is still increasing up to more than 115 characterized in 2022. But this is in fact a very old class of natural products. And the example is nisin, which is a food preservative that you probably already saw in the labels of food that you buy in the supermarkets, with a E234. And this compound was discovered in 1928 together with penicillin, but only in 1947 it was completely characterized. Until now, lots of lantipeptides have been discovered, and their activity is being explored and characterized, and of course the structure of these compounds is also being elucidated. But nisin it's an example for us because it has been used for more than 60 years now, and no significant incidence of microbial resistance has been observed. And this is probably due to the mode of action of these compounds. They have a dual mode of action, which is different from the traditional antibiotics. It interacts with lipid-2, which is a component that is involved in the synthesis of the cell membrane of bacteria. And it also places itself within the membrane, forming some pores, which lead to bacterial lysis. So the bacteria has in fact to deal with two mechanisms and two modes of action. So usually these compounds have low tendency to generate resistance, and they also very low toxicity. So they are very attractive for medical applications. Regarding the biosynthesis mechanism of these compounds, as I told you before, they are synthesized as a precursor, which is quite long and has a core peptide and a leader peptide. And this structure goes through a number of post-translation modifications, namely dehydration, which leads to dehydroxybutyrene and dehydroxyalanine. And then a second post-translation modification, which is a cyclization, which occurs by the formation of these thioester bridges that occurs between the cysteine, the diol group of cysteines, and the amino acid. And it gives rise to these polycyclic structures that are, of course, very resistant to heat and also to proteases, which is one of the problems of the compounds and antibiotics, is that they usually are degraded by proteases, and they have proteolytic activity that occurs in the organ in our body. And also they are very specific in their target. So this is one of the characteristics that is attractive of these lantipeptides. After they have been synthesized and cyclized, the leader peptide is removed, and only now the peptide is able to traverse the membrane and become active on the outside, on the supernatant. Okay, so for this, of course, the bacterium must have some immunity genes that makes it resistant to its own compound, and I will talk about that in a minute. The biosynthetic machinery of these antipeptides, there are a number of enzymes that are involved in these post-translation modifications. I'm not going into much detail because some of you don't have, the background, the adequate background for this, but this class 1 is particularly important because I'm going to talk about it a little bit more. And these enzymes, the dehydrase and the cyclase, are involved in this in the post-translation modifications as I showed in the previous slides. So this is an important class because, for example, nisin belongs to this class of compounds. The genes encoding lantipeptides, or the enzymes that are involved in the synthesis of lantipeptides, they have been found in all the three domains of life. In bacteria, they have all the four classes. In eukaryotes, there is a fifth class that has been discovered recently, and also in archaea, they have probably usually class 2 arrays of enzymes. So far, only in bacteria these lantipeptides have been characterized and isolated. So in archaea we are working on that in my group, and in eukaryotes there's not too much work done so far. One of the advantages of these lantipeptides, and particularly the RIPs, the ribosomal encoded peptides, is that there is a direct link between the gene and the final peptides. So we can do genome mining, so we can look in the genomes for the enzymes that are involved in the synthesis of these peptides. We can make structure activity studies like site-directed mutagenesis, or we can do bioengineering, directed evolution. So we can create novel molecules with improved activity, with specific targets, and so on. So if you're not a geneticist, it's going to be difficult to understand this, but this is a biosynthetic gene cluster encoding a peptide, for example, licanicity. We have the modification enzymes here characterized by M1 and M2. We have the core peptide. We have the transporter to transport the peptide across the membrane, usually ABC transport systems. We have the protease. We have the regulation genes that are involved in the regulation of the synthesis of the peptide, and we have also the immunity genes that are involved in protecting the bacteria from its own peptide once it is secreted. So you can see also that there is a long precursor peptide. Here is only the core peptides, which is the only thing that is that has activity, in fact. And after these, the post-translation modifications, the leader sequence is removed, so all of this is removed, and only the peptide, the final peptide, is produced. Okay. For example, here, this peptide, or these lantipeptides, is a two-component lantipeptides. As you see, there is a core peptide A1 and A2, and only when we have both peptides interacted, so in a synergistic effect, we have bioactivity. For itself, individually, they don't have most activity. Blibeta has some, but only when they are together is the maximum of their activity, so a synergistic effect. When we do genome mining approaches, we look for the signatures or identities of these enzymes that I've talked about, and we can see if a microorganism has the potential to produce new compounds. Usually, these biosynthetic gene clusters, they are in the genome, but they are silent. And when we want to wake up these genes, we have to introduce them in a host that is going to express those genes, and then we can do studies with these peptides. So this is a gene to compound connection. We can also do the other way around. We can identify the compound and then go back to the genes. So in my group, we work with two lantipeptides, one produced by a gram-positive bacteria and another one produced by gram-negative bacteria. And we also have some work done in archaea. Okay, so just to give you an example, without, I don't want to bore you too much with this issue, but we have isolated the bacteria from an extreme environment. We sequenced its genome, and using bioinformatic tools, we saw that in its genome, it has 17 biosynthetic gene clusters. among them, there are 6 biosynthetic gene clusters for lantipeptides. And we notice that all of them are class 1 lantipeptides, so like nisin. So here you can see that in orange, there are the LAN-B enzymes, which are the enzymes that are involved in part of the modification. And we also have a transporter, so we have the range of all the genes important for the expression of or the synthesis of a peptide. So we selected these clusters because, as I told you before, it's possible to predict the sequence of the peptides because this is a genome-encoded peptide. So we predicted the amino acid sequence, and when we looked at all these peptides that were supposedly produced by bacteria, but we cannot detect them, so they are silent, we wanted to know why they are not produced in the bacteria. So they all have the machinery required to do those post-translation modifications that I've talked in the beginning. So we started with one of them, pepkins, and we wanted to see what was the dehydration pattern, how it occurred. So what we did, as the peptide was not being produced in the original host, we had to do heterologous expression in E. coli. And that was one of the good things that we did because it was very important. It broke a dogma of the biology. We could express gram-positive genes from a gram-positive bacteria. We can express and have compounds with bioactivity in the gram-negative host E. coli. So we introduced all the genes necessary to have the peptide. We introduced an E-style to help in the purification of the peptide. We did mass spectrometry, and we could not find the expected mass because there was no dehydration. So we wanted to now understand why this was happening. And we were aware that in nisin, which is a class 1 peptide like pepkins, the mechanism of dehydration of serines and threonines is different from the other peptides. It uses a tRNA glutamylated, which transfers by esterification reactions a glutamate for serine and threonine. So the first step of the dehydration process is a glutamylation, and after that the glutamate is removed and the amino acids are dehydrated. So the process of dehydration is different from, for example, class 2 peptides, but we heard about that in nisin. So we wanted to know if that was the reason why we didn't have dehydration and we could not detect any peptides. So when we did this system without the tRNA for glutamate, we didn't have dehydration. But when we added a glutamyl tRNA from E. coli, we can see dehydration, but no bioactivity still. So we could detect peptides, but they were not bioactive. So we are not now working on this subject, trying to understand why the peptides are being produced, but they are not still bioactive. So we are still working on that. So I'm not going to go any further in this part, but I wanted to show you that there are lots of lantipeptides that are already in phase 1, 2, or also reached final clinical trials. And here you have some examples of them. The major problem we have also is the yield, because we need high amounts, and usually this is not obtained in the original producer. So usually we have to do heterologous expression, and then we have to do other manipulations to have high yields of compounds that might be interesting for the industry. So here you can see some of them are already being commercialized. For example, these two for MRSA, for vancomycin-resistant enterococci, for Clostridium, and another example of a compound that is used for cystic fibrosis. And these are only some examples of compounds that are already in use or in final clinical trials. Nisin undoubtedly was the one of major success. It's been produced and used for many years, and now is being studied for its anti-tumor activity, which is quite exciting. As I told you before, and as I showed you, for the peptides to become active, they undergo these post-translation modifications, and the applications of this biosynthetic machinery is being used to design novel therapeutic compounds. And there are some companies that really developed their drugs based on enzymes from lantipeptides, namely LAN-M and LAN-B, which are involved in improving activity, in increasing the spectrum of activity of the compounds, and also in peptide stability. And here are some examples of some peptides. Some of them are hormones. And one example of success is this angiotensin that you probably know that is involved in high blood pressure. Angiotensin 1 is converted in angiotensin 2 by angiotensin converting enzymes. And when this conversion takes place, high blood pressure occurs. So one way of diminishing this high blood pressure is using compounds that have been manipulated using these enzymes that avoid the degradation by the peptidases. Okay, so MOR 107 is an agonist of the AT2 receptor and is a compound that is commercialized for high blood pressure. And Lantio Pharma is the company that develops these lantipeptides with interesting therapeutic application involved, for example, in cure of diabetes, cardiac diseases, and hypertension. Also, the proteases are other enzymes of these lantipeptides are also being used in the industry. As you know, proteases are very, very important in various sectors of the industry. And the use of these enzymes can improve the flexibility of these compounds, can help in the tag removal for specific applications of some compounds, and also in the peptide solubilization and transport of also other molecules. And one example that we did in our lab, in my lab, it was with insulin. We used a LAN protease to in vivo break the insulin and make it active. So this is also an example of application of the proteases, of the machinery of these antipeptides. So to finish, I just want to tell you that we are in the tip of the iceberg. So many lantipeptides are still to be discovered. The application is enormous in different areas, as you see, and also the compounds itself, the peptides itself, they have many applications. So all this work also was done in collaboration with some colleagues from the United States and also from United Kingdom and Israel. So just to present you the team, the LBM team, most of the students don't work in these peptides specifically, but some of them did some work here. So I would like to thank to them also for their work, and thank you very much for your attention and for your kind invitation. Thank you.
Ok, muito obrigado, professora Sônia, pela sua apresentação, por compartilhar a sua pesquisa conosco. Pedir agora,
Obrigada.
por gentileza, que interrompa o compartilhamento da tela.
Sim.
Aí.
Ok, muito obrigada.
Obrigado. Então, daremos continuidade agora com a fala da professora Mariela Xereg. A professora Mariela atualmente trabalha nos Estados Unidos e não pôde estar presente aqui para fazer a sua apresentação ao vivo, mas nos enviou um vídeo com a gravação, então, da sua fala, que será apresentada a partir de agora. O título da fala é La investigación artística en el cruce arte, ciencia, tecnología.
Buenas tardes. Antes que nada, quiero agradecer esta invitación. Me parece una oportunidad excelente el hecho de estar compartiendo con ustedes mis pensamientos, mis reflexiones en torno a estos diálogos, a estas interacciones entre el campo artístico y la ciencia. Y bueno, quiero agradecer a la Universidad de Santa María por abrir estos espacios que son sin duda espacios de mucho crecimiento y muy nutricionales. Yo voy a hablar de el cruce entre arte, ciencia y tecnología y sobre todo el cruce de estas disciplinas dentro del campo del arte tecnológico y cómo la investigación, qué forma cobra la investigación en este campo. Para ello voy a empezar con esta imagen, que es la imagen de un proyecto del arquitecto Claude Ledoux, un arquitecto del periodo iluminista del siglo XVIII, quien junto a Etienne Boulet formaba parte de lo que se denominó la escuela visionaria, ¿no? Y Ledoux diseñó una ciudad ideal, una ciudad que desafiaba los presupuestos del clasicismo, poniendo en jaque incluso a la propia ley de gravedad. Al estilo clásico, que recordemos estaba regido por el orden y la sujeción a los cánones, a la simetría de las proporciones, al equilibrio entre las partes, a la distribución rítmica de las masas, Ledoux opone la no sumisión a las reglas preestablecidas, la autonomía de las partes y de las formas, el juego de las masas y la posibilidad de que cada material actúe de manera independiente. Como señala Kaufman en su libro de Ledoux a Le Corbusier, Ledoux reemplaza el sistema de encadenamiento de partes por el de libre asociación de unidades autónomas. Este concepto de libre asociación nos dará las claves para entender la cuestión de lo transdisciplinar y en particular de qué se trata la investigación artística en el cruce arte, ciencia y tecnología. Pero es importante primero diferenciar tres términos que por su semejanza a veces producen equívocos en el nivel de sus significados. Me refiero concretamente a los términos pluridisciplinar, interdisciplinar y transdisciplinar. Para ello seguiré la teoría de Basarab Nicolescu, uno de los pilares del enfoque transdisciplinar. Entonces, una perspectiva es multidisciplinaria cuando estudia un objeto a través de diferentes disciplinas. El objeto es el mismo, pero los abordajes son diferentes. Es un enfoque esencialmente aditivo. Por su parte, lo interdisciplinar concierne a la transferencia de métodos de una disciplina a la otra. Esta transferencia sucede no solamente a nivel de la aplicación esta metodología, ¿no? Cómo se la aplica, sino también en el nivel epistemológico. Es decir, un método de una disciplina transferido a otra puede generar consecuencias epistemológicas en la disciplina receptora. Y también puede afectar al nivel conceptual. Es decir, un método de una disciplina transferido a otra puede hacer surgir nuevas disciplinas. Entonces, el enfoque interdisciplinar es un enfoque que privilegia la intersección. Lo transdisciplinar, por su parte, y acá traigo esta imagen de un gran rompecabezas, un puzzle, porque me parece una imagen atinada para dar cuenta de esta mecánica. Lo transdisciplinar, decía, alude al espacio entre, a través y más allá de las disciplinas. Por eso puse este puzzle y vemos que el objeto de estudio, que es este círculo negro que está por detrás, está constituido justamente por cómo las diferentes partes se van vinculando, o sea, las partes van construyendo a este objeto. Entonces, en virtud del carácter múltiple de la realidad, en virtud de esta simultaneidad de niveles, de la complejidad y de su estructura discontinua, o sea, no lineal, la hiperespecialización que el conocimiento experimentó durante casi todo el siglo XX entra en contradicción con la naturaleza múltiple de lo real. De esta forma, el enfoque transdisciplinar de alguna manera zanja esta contradicción, proponiendo la integración o diálogo entre las disciplinas, pero además va más allá, porque propone un viaje integrador por este universo diverso del conocimiento. La conciencia transdisciplinaria permite entonces tender puentes que rompen la mirada compartimentada y liberan así al sujeto transdisciplinar, al sujeto que emprende este viaje transdisciplinar, lo libera de los particularismos que imponen los guetos del conocimiento. Niels Bohr, uno de los máximos exponentes de la física cuántica, ha sido de los primeros en referirse al concepto de transdisciplinariedad. A este respecto, Bohr afirma, cito a Bohr: el problema de la unidad del conocimiento está íntimamente ligado a nuestra búsqueda de una comprensión universal. Se trata de comprender de manera armónica los aspectos cada vez más vastos de nuestra situación como humanos. Entonces, el principio de complementariedad que enuncia Bohr, que se basa en la dualidad ondulatoria y corpuscular de los cuerpos, completa esta visión inclusiva de lo real. Frente a la cuestión de si la luz era movimiento ondulatorio o corpuscular, Bohr observó que en realidad reunía ambos caracteres al mismo tiempo, en simultáneo. De esto se desprende que para Bohr dos realidades o dos aspectos de una realidad que han sido considerados como opuestos en la física clásica pueden ser complementarios para la física cuántica. Esto lo vinculo también con la teoría del sociólogo francés Edgar Morin, que enuncia el principio del tercero incluido. Frente al principio del tercero excluido de la lógica clásica, según el cual una cosa puede ser o no ser, pero no hay una tercera posibilidad, Edgar Morin nos dice que es posible la inclusión de un enunciado contradictorio y que esto puede ser visualizado como algo complementario. O sea, ambos enunciados conviven, solo que en diferentes niveles, se traduce por ejemplo, la afirmación yo soy, no soy. Esto sería perfectamente válido según el pensamiento de Moren, y esto expresaría esta idea de tercer excluido, ser, no ser, ¿no? Ser lo negativo. O sea, ambos están incluidos, ambos conviven, y por eso es el principio del tercero excluido. Incluido, perdón, no excluido. Entonces, como decía hace un rato, la transdisciplinariedad propone examinar estos mecanismos de interconexión entre las disciplinas. Estos espacios que las separan a las disciplinas no son espacios vacíos en términos clásicos, sino que constituyen un vacío desde una perspectiva cuántica. En el vacío cuántico, los pares virtuales de partículas y antipartículas se crean y aniquilan continuamente. Es un vacío plagado de actividad y de potencialidades, a diferencia del vacío ordinario definido por la ausencia de materia y por un estado de reposo. Entonces vemos que el universo de la transdisciplinariedad supone la emergencia de las siguientes dinámicas y principios. 1. Lógicas inclusivas en las que pueden convivir paradigmas divergentes. 2. Sistemas relacionales que desbaratan la parcelación del conocimiento, privilegiando, por el contrario, la retroacción, la interacción, la interconectividad, en un entramado complejo marcado por lo multidimensional. Y tercero, la discontinuidad como principio de organización de lo real. En tal sentido, el conocimiento debería dar cuenta de este carácter no lineal de la realidad, promoviendo enfoques que se imbriquen unos con otros y que se retroalimenten y se intercomuniquen. El epistemólogo Karl Hempel, aludía a esto cuando hablaba del carácter multidimensional de la ciencia, y lo cito: la sistematización científica requiere el establecimiento de diversas conexiones mediante leyes o principios teóricos entre diferentes aspectos del mundo empírico que se caracterizan mediante conceptos científicos. Así, los conceptos de la ciencia son nudos en una red de interrelaciones sistemáticas en las que las leyes y los principios teoréticos constituyen los hilos. Cuanto más hilos converjan o partan de un nudo conceptual, tanto más importante será su papel sistematizador o su alcance sistemático. Entonces, ¿por qué deberíamos suponer que el hecho de hacer resonar la cuestión de lo transdisciplinar en el campo artístico pueda ser un desarrollo pertinente? ¿Y cómo ha repercutido esta línea de pensamiento en el campo de las artes y en particular en el campo de las artes tecnológicas? Las artes tecnológicas, es decir, aquellas prácticas que incorporan los medios de tecnología analógica o digital como lenguajes de expresión, suponen un replanteo de los paradigmas estéticos tradicionales y una revisión de las posturas academicistas y ortodoxas en relación al arte, y el comienzo de un diálogo entre la práctica artística y el desarrollo tecnológico. Ya a principios del siglo XX, las vanguardias históricas, es decir, Dada, Futurismo, Surrealismo, Bauhaus, entre otros, manifestaron un interés marcado por la incorporación de principios técnicos y mecánicos, articulando entornos multi e interdisciplinarios en la creación artística y reformulando el rol del creador y del espectador en la situación comunicacional. Todos estos que menciono son rasgos esenciales de las artes tecnológicas de la actualidad. A modo de ejemplo, el biólogo von Wexkull, una figura central que influyó a muchos científicos y filósofos. Entre los filósofos menciono a Heidegger, a Maturana, Merleau-Ponty, bueno, Maturana, científico también, Merleau-Ponty, Foucault, Deleuze, Guattari, Agamben, entre otros. Pero también influenció artistas como por ejemplo Moholy-Nagy y sus formas biotécnicas, o el proyecto utópico de sistema reconstructivo cinético de Moholy-Nagy, que se inspira en el concepto de biotécnica del biólogo Raoul Francet, uno de los referentes de la Bauhaus. Entonces estaría tentada a decir que las artes tecnológicas surgen en estas zonas intersticiales que conectan a disciplinas múltiples. Las artes tecnológicas llevan la transdisciplinariedad como una marca indeleble en su estructura epistémica. Cabe también recordar que uno de los campos de conocimiento que ha impreso un impulso vital, fundamental, en la génesis de las artes tecnológicas es la cibernética. También recordemos que es una corriente sustentada por los desarrollos de científicos tales como Norbert Wiener en la década del 40, y que la onda expansiva de la cibernética irrumpe en el escenario artístico de la mano de científicos, y no de artistas, sino de científicos, que comienzan a explorar la arquitectura de las máquinas de información con el objeto de generar experimentos de creación. A este interés inicial por la relación hombre-máquina se sumaron diversos artistas y científicos, ya sea para operar sobre el aparato, o sobre el hardware, o bien para generar reglas e instrucciones que posibilitaran la creación aleatoria y o automática, y así actuar a nivel de la inteligencia de la máquina. Entre estos personajes menciono a alguien que fue central, que es Frank Malina, una curiosa amalgama de artista e ingeniero. En la década del 40, Malina se consagra a la concepción y desarrollo de cohetes espaciales, y poco más tarde realiza trabajos artísticos de corte cinético. Malina sintetiza en cierta forma las dos vertientes por las que se cristalizaba la cibernética dentro del ámbito artístico. Por un lado, Malina diseña su propia aparatología para generar estas obras. En ese sentido, el hardware no solamente una instancia que interviene en la creación, sino también es el propio soporte de exhibición. Como la obra involucra aspectos dinámicos y un devenir temporal, la obra se crea al mismo tiempo que se exhibe, se crea mientras se exhibe. Y es lo mecánico también un elemento protagónico tanto en la creación como en la exhibición. Malina anticipa así un rasgo que luego cobraría impulso en las artes tecnológicas. O sea, la obra como un sistema dinámico, las instancias de creación y exhibición que terminan indiferenciándose, emergiendo así el carácter procesual de la obra como uno de los rasgos protagónicos. Una de las iniciativas de Malina fue la creación de la revista electrónica teórica que discute todos estos temas, que es la revista Leonardo, que hasta el día de hoy sigue activa, una revista de referencia editada por el MIT, Massachusetts Institute of Technology. En esos mismos años, mediados del siglo XX, uno de los personajes más influyentes es Billy Kluver, un ingeniero eléctrico que alterna su trabajo entre la compañía Bell y colabora a su vez con artistas. Además de trabajar con Jean Tinguely en la instalación cinético-mecánica Homenaje a Nueva York, también aporta sus conocimientos para la creación de la instalación Nubes de Plata de Andy Warhol, que era una serie de rectángulos metalizados rellenos de gas helio flotando en el ambiente. También colabora en la resolución de la alimentación por baterías de las letras de neón de la pieza Field Painting de Jasper Johns, y también participa en la pieza sonora Oráculo de Robert Rauschenberg, y asimismo colabora en obras de danza e instalaciones sonoras de Yvonne Rainer, John Cage y Merce Cunningham. Pero además es curador de varias muestras, alternando su trabajo con como ingeniero en la empresa Bell. Entre las exposiciones está Popular Image en la Washington Gallery of Modern Art, también American Pop Art en Estocolmo. En 1966, junto a su compañero de trabajo Fred Walkhauer y junto a los artistas Robert Rauschenberg y Robert Whitman, funda el grupo Experiments in Art and Technology, EAT, el cual se consolidó como un ámbito consagrado a la investigación transdisciplinar y al quehacer colaborativo. EAT proclamaba lo siguiente, cito: mantener un clima constructivo para el reconocimiento de las nuevas tecnologías y las artes a través de la colaboración de los grupos que trabajan absurdamente en aislamiento. Eliminar la separación del individuo del cambio tecnológico para otorgarle variedad, placer y avenidas de exploración y participar en la vida contemporánea. Estimular la iniciativa industrial para generar proyectos originales en lugar de transigir en función de los desenlaces y activar acuerdos mutuos para evitar el fracaso de una revolución cultural. Entonces, bueno, este grupo EAT se constituye en un referente del trabajo transdisciplinar y también de la aproximación desde el punto de vista metodológico, de la aproximación transdisciplinar, en tanto es un enfoque a una realidad múltiple a través de metodologías abiertas y exploratorias. Entonces, podría decir que el carácter transdisciplinar de las artes tecnológicas se visualiza a partir de su propio nacimiento y génesis. Entre los intersticios que separan las dos culturas de las que hablaba Joe, las artes electrónicas o tecnológicas crean bisagras que permiten la intersección arte-ciencia. Entonces, las artes tecnológicas articulan un impulso transdisciplinar en la propia definición de su campo artístico. En ese sentido, el arte tecnológico configura un objeto dialógico que aborda una pluralidad de lenguajes, encaramándose incluso en el terreno de lo híbrido, de la hibridez discursiva y estética. Pero a su vez, este dialogismo se manifiesta en el hecho de que las obras proponen una experiencia multidireccional. El concepto de autor se diluye para dar paso a una multiautoría que surge de esta posibilidad de que la obra es actualizada permanentemente por cada espectador. Es sin duda, uno de los rasgos más salientes del arte tecnológico, es decir, el hecho de poner en el centro de la discusión al entorno y al espectador, haciendo entonces del artista un facilitador de contextos más que un creador virtuoso o inspirado. Entonces es innegable que el objeto ya no es un objeto cerrado, sino un objeto en permanente transformación. De igual manera, el abordaje transdisciplinar sustenta como principio de acción el cruce de voces y miradas en la constitución del objeto científico. De esto se desprende que el arte tecnológico tiene una orientación transdisciplinar en su propio sustrato conceptual. A su vez, surge en este campo artístico una práctica que es sintomática de este concepto transdisciplinar, que involucra la idea de redes, de interconexión, de nodos como puentes entre canales y fuentes de información diversas, ¿no? Recordemos esa imagen de los hilos que mostré hace un rato. Y como ejemplo de estas interconexiones, voy a tomar el caso de la metáfora biológica de las máquinas, que de alguna forma da cuenta de estas colaboraciones, contaminaciones y diálogos que operan en la investigación artística en las artes tecnológicas. Y bueno, y traigo la figura de Turing, de Alan Turing, ¿no? Y recordemos que este matemático en 1948 publica el artículo Intelligent Machinery, y ahí describe los diferentes tipos de máquinas computacionales, entre las cuales se encuentran las máquinas tipo B, a las que se le incorpora la capacidad de aprendizaje a partir de la conexión entre neuronas, lo que comúnmente llamamos sinapsis y que anticipa lo que más tarde se dio en llamar redes neuronales, algo que en los tiempos que corren está en la cresta de la ola si pensamos en los sistemas de machine learning e inteligencia artificial. Obviamente están fundados en estos modelos matemáticos fundacionales, ¿no?, de la computación. Pero lo interesante es que también en estos modelos surge la retórica de lo vivo, ¿no? O sea que lo biológico, lo computacional, lo artístico, impregna el lenguaje de las máquinas, ¿no? Y esto es fundamental en un tipo de lenguaje artístico que trabaja con máquinas, como es el lenguaje de las artes tecnológicas. Es así que nociones tales como dialogismo, no linealidad, multidimensionalidad, muy estrechamente vinculados con la perspectiva transdisciplinar, aparecen en el dominio conceptual de esta práctica artística. Lo que es importante enfatizar es que esta relación ciencia, arte, tecnología se produce desde los intersticios que la transdisciplinariedad pone en escena. Se trata de una dinámica transdisciplinar la que genera este objeto, ya que es a partir de estos vacíos cuánticos de los que hablábamos al inicio, esta zona de circulación entre las disciplinas, es ahí que el objeto artístico tecnológico nace. Nuevamente, citando al Edu, la libre asociación de unidades autónomas sobre la que se sustenta la arquitectura del Edu pareciera estar preanunciando la noción de imbricación de múltiples niveles, tal lo que postula la corriente transdisciplinar. Más de 2 siglos más tarde, el transarquitecto Marcos Novak enunciaría el concepto de transvergencia, que yo creo que es muy cercano a la noción de transdisciplinariedad. Cito a Novak, dice: mientras que la convergencia y la divergencia están aliadas a epistemologías de la continuidad, la transvergencia está epistemológicamente más cerca de la lógica de teorías relacionadas con la incompletud, la complejidad, el caos y la catástrofe, los sistemas dinámicos, la emergencia y la vida artificial. Mientras que la convergencia y la divergencia enmascaran la asunción de que la verdad, tanto en un sentido cultural como objetivo, es una masa continental continua, la transvergencia reconoce proposiciones verdaderas como islas en un archipiélago foráneo, a veces solo accesible a través de saltos, vuelos y viajes por las tuberías del artificio. Para cerrar, quiero decir que los derroteros de lo transdisciplinar y del arte tecnológico, en tanto caja de resonancia de la transdisciplinariedad, están marcados por un fuerte nomadismo. nomadismo intelectual, cognitivo y creativo de la instancia creadora, artistas, ingenieros, científicos, teóricos. Un sujeto nómade que puede caminar a la deriva por este mapa dinámico del conocimiento. Muchas gracias.
Ok, agradecemos então a professora Mariela Xereg pelo envio da sua fala. Faremos um pequeno intervalo de 10 minutos até a conferência final do professor Henning. Então, voltaremos em 10 minutos.
Estamos ao vivo.
Ok, retornamos então agora para a conferência final do Sinergia. Rosa, passo a palavra para ti.
Welcome, Professor Henning. You have up to 40 minutes for your lecture, Cholinergic Signaling in Neurodegeneration. We kindly ask you to start your speech with your professional presentation, and please feel free to start.
Professor Hauser, thank you very much for this very kind invitation. So I'm always very honored to come to Santa Maria and to give my lectures, and also I'm very honored by this online lecture. So now I will share my I will now share my screen. And start my lecture. So, thank you very much again for inviting me to give this lecture. And my lecture today will be about the modeling of neurodegeneration and cholinergic signaling in vitro and also in vivo. So the first work which I will talk about will be a study in vitro, how we can use stem cell models to model neurodegenerative diseases. And for this, we use stem cells which are modified by expressing mutant proteins, or we use the stem cells from patients, for example. And then we will treat with stem cells during the differentiation with drugs and to see what is the impact of cholinergic signaling. And here I will come a little bit more to this topic. So these are stem cell models which we use in my laboratory, where we use neural stems and progenitor cells, and these cells, they can be obtained by primary cultures of, for example, of neurospheres, which are from a telencephalon of the rat or mouse embryonic brain, or we can use stem cell lines, like embryonic stem cell lines, and pre-differentiate them into neural stem and progenitor cells. Or it is also possible to use embryonic carcinoma cell lines, or what has been put very much, what has been done very much now is to use induced pluripotent stem cells, which are iPS cells. So now the question is, how well these stem cell models reflect neural development and neural network. And what you see here in this slide, this is an embryonic stem cell line which has been induced to differentiate into neurons. And what you see here, a very nice labeling of a neural network by a marker, TOUJ1, which labels young neurons. And what you see here in blue are the cell nuclei. So now, the first work, which I will show for you, that we used for guiding neuronal differentiation and modeling for the differentiation into GABAergic neurons, is this mouse embryonic stem cell line. And here you see, after 4 days of induction of differentiation using retinoic acid and suspension culture, you will see that these cells were yet express a little bit of OCT4, which is marking their pluripotency. But at this stage already, they mark for stem cell specific embryonic antigen 1 and also for nestin. It means that the differentiation goes into the lineage of neural stem and progenitor cells, where we have a maximum on 4 days. Then on day 8, we have a presence of young neurons marked by beta-3 tubulin, and we have also glial cells, so marked by GFAP, and oligodendrocytes marked by GAL-C. After 16 days, the differentiation is complete. As you can see, they express major astrocytes, major neurons, and basically, which would be the end of differentiation. So now, the first work, using this cell line, I will show you a little bit about the idea that ATP drives neural differentiation. And differentiation is always well aligned also to neurodegeneration. So if we understand neurogenesis, then we also understand a lot about neurodegeneration. So this first work will show how extracellular ATP drives neural differentiation. And just before I go into details, I want to explain you how works purinergic signaling. So purinergic signaling is induced by nucleotides like ATP and other nucleotides. And what happens? But with nucleotides, ATP are normally very well known for their functions inside of the cell, where they act as energy stores and for catalyzing reactions in the cell. So now, but also ATP is being released by several mechanisms into the extracellular space, so by channels, by exocytosis, or by leakage of the membrane when cells die, yes? So now, with ATP that you have outside of the cell, which will activate P2X and P2Y receptors. P2X receptors are ion channel, which are assembled by three subunits, while the P2Y receptors are typical G-protein coupled receptors, and they will be activated by ATP and other nucleotides, and they activate a G-protein coupled signaling. And many times they also are inducing ion currents. But I forgot to say that with ion channels, so that let the inflow of sodium and calcium ions and the outflow of potassium ions. Okay, so outside of the cell, the ATP will by ectonucleodases be degraded into adenosine, and adenosine is activating P1 adenosine receptors. So in many concepts, P2X, P2Y, and contrasted to P1 receptors may have antagonic functions. For example, in the immune system, where the ATP is more pro-inflammatory, while adenosine would be anti-inflammatory. So now, here you see the diversity of purinergic receptors. I won't go into any details about adenosine receptors, but I will focus here on P2 receptors, which are activated by ATP and other nucleotides. And I will, on the left side, you see that there are 7 subunits for P2X receptors, which can be assembled as homomeric or heteromeric receptors, while P2Y receptors, they are activated. They are activated, and they also by ATP, UTP, and other nucleotides, and they have in mammalians 8 subtypes. And I will here focus on 2 receptor subtypes, that is the P2Y2 and the P2X7 receptor. And what I propose here to you, that they have antagonic function with 2 receptors. So now we see that that embryonic stem cells, that they differentiate into neurons and major glial cells within 16 days, what I showed you with embryonic stem cell line. And what is here calling attention is that after, when cells become totally differentiated, when also the extracellular ATP concentration declines. And this says, okay, maybe there is a participation of polynergic receptors in neural differentiation. And what you see here, in this slide here, that if you look at the differentiation, so if you do live cell imaging, that means you will culture the cells on the microscope and let them differentiate, and you look here for intracellular calcium fluxes using a fluorescence probe. Then you will see that you have here an oscillatory behavior. So you have calcium oscillations, and the hypothesis is here that very low ATP concentrations and other neurotransmitter that they continuously activate these receptors, and their concentrations are too low to induce any receptor desensitization. Okay. Now, the idea is that this extracellular calcium, yes, through an influx through P2X receptors would cause spike activity of calcium inside the cell, while intracellular calcium stores released through the endoplasmatic reticulum would cause with slower calcium oscillations, which are caused here waves. Okay, so it means that P2X receptor would be responsible for spike activity, while P2Y receptors would be responsible for wave activity. Okay, so this is all fine. Since I said I will talk about neurodegeneration, I don't show you here a big number of experimental details. What I would show you here, if you use pluripotent stem cells, embryonic stem cells, and we screen for receptor activity, then we stand out two receptors which we will investigate here in the following. So one of them is the P2X7 receptor, which maintains proliferation of pluripotent stem cells and events differentiation. And then is the P2Y2 receptor. This is a receptor needed for the differentiation of pluripotent stem cells into neural progenitor cells and then to mature neurons. On the other side, when we maintain the P2X7 receptor, then we would have differentiation into glial cells. Okay, that means for neurogenesis, for enriching neurons, we will need to downregulate or inhibit the P2X7 receptor. Okay, so this slide here now shows the ongoing differentiation of these embryonic stem cells. And what stands out here, that these cells can be differentiated into GABAergic neurons, yes, shown by immunolabeling for GAD65, 67, yes. You see this, but the neural progenitor cells has already GABAergic markers, and then we get much more often when these cells are finally differentiated. So now, okay, we say, okay, maybe we need polynergic receptors, we need ATP binding to cell surface receptors, and we need fluxes of intracellular calcium and calcium influx to trigger the progress of neuronal differentiation. And what I show you here is a scheme, just looking a little bit of this. So ATP released from neural progenitor cells through pannexine channels, for example, it will bind to P2Y receptors, and such as the P2Y2 receptor, and to P2X receptors, such as the P2X7 receptors. There's also participation of voltage-gated calcium channel, which I won't detail here. But what happens inside the cell is after activation of a GK protein, we have the release of intracellular calcium from the endoplasmatic reticulum, and this calcium binds to kinases, activates kinases, for example, like calmodulin kinase. And this kinase activity will then finally generate the activation of neural transcription factors. And these neural transcription factors here, which stand out, are two of them. So it's MASH1 that is known to trigger neurogenesis, really. And then there is neurogenin 2, which stands out because it's more or less inhibiting the progress of neurogenesis. And basically, the idea is that this differential activation of these neurotranscription factors will basically trigger the progress of neurogenesis. Okay. Now, the next thing here is, now we have, we know that is MASH1, which is basically the idea here is that it has an oscillatory expression behavior. And when MASH1 expression becomes sustained, it would trigger neurogenesis. And what we want to do now, that we want to put together a spike and wave activity that is also oscillatory to oscillatory neural transcription factor expression and the progress of neurogenesis into GABAergic neurons. Okay, so now for this, we used a transgenic mouse line, yes, for bioluminescence time-lapse imaging. That means what has been done to a luciferase promoter together with one of these transcription factors. So if I induce expression, I will have a luciferase transcription and expression, and I will be able to measure luminescence. Okay, so we were done this by microinjection with mouse models, transgenic mice, and then basically the breeding of these transgenic mouse lines, and then the extraction of embryonic stem cells. And these embryonic stem cells then can be used for differentiation and monitoring of MASH1 and neurogenin 2 gene expression in lifestyle imaging. It means that we have here the idea that we may look at a biological clock so that we have activities which happen here in seconds and put them in connection with activities which happen here within hours or days, like gene expression. Yes, so it means what I show you here on the right side again is the calcium oscillation that I had shown you before, and here on the bottom I show you here this is MASH1 gene expression oscillation. You see the time scale is different. This is in seconds, this is in hours. Okay, but we can achieve this to put this together using time-lapse imaging. So in the next slide was again the justification why we were looking at P2X7 and P2Y2 receptors. And what you see here, the P2X7 receptor, as I was telling you before, was that with P2X7 receptors are getting downregulated in their expression following induction of differentiation to neurons. Yes. On the other side, the P2Y2 receptor gets upregulated at the peak of neurogenesis when young neurons appear. And then differentiation is complete, the P2Y2 receptor will be downregulated. And here we do the experiment where we match calcium imaging, calcium transience oscillation, and here on the right side, we put together the activation measured by luminescence of mesh 1, okay? And what we can see here, that the control that you see on the top naturally will induce at some stage mesh 1 expression. It becomes sustained and cells will differentiate. On the other side, now if we interfere with calcium signaling by inhibiting voltage-gated calcium channels, yes, or by blocking the entry of extracellular calcium, or by depleting intracellular calcium, you see, but this all abolishes the activation of mesh 1, okay? On the other side, if we look here at 2S-UTP, that in mouse is a specific agonist of the P2Y2 receptor, we see an immediate activation of mesh 1 transcription, and if we use an agonist of the P2X7 receptor, then we won't get any activation of mesh 1. And now, if we do the same experiment, yes, neurogenin 2, when we see just that there is an opposite action for P2X7 receptor agonist of BC-ATP, which induces neurogenin 2, and the P2Y2 receptor agonist, 2S-UTP, would not induce the expression of neurogenin 2. So I mean, we have opposite actions by these two receptors on these neural transcription factors. So now, going on with the story. So you see the 2S-UTP is the only one is used conditions which really induces the expression of GABAergic markers, saying that with neural stem cells differentiate into GABAergic neurons. While the other conditions, BC-ATP activating P2X7, maybe other P2X receptors, is just increasing a little bit here of cholinergic receptor expression. But it does not induce any other expression. And you see here, the expression for GABAergic neurons is much lower. So what we're asking here was for glutamatergic neurons, for dopaminergic neurons, cholinergic neurons, serotonergic neurons, and GABAergic neurons, okay? And now, if we now measure the ratios between MASH1 and neurogenin 2, when we see that 2S-UTP increases as expected with ratio. So there's much more MASH1 activated than neurogenin 2, while BC-ATP activating P2X7 receptor is doing the opposite. And now this means that we have a scheme that we could follow, so it was following our hypothesis. So the idea is that P2Y2 receptor would induce calcium fluxes through the endoplasmatic reticulum, and with calcium oscillations, they induce calmodulin kinase, they would induce PrEP phosphorylation, and finally, they would activate MASH1 transcription and basically the increase of neurogenesis, yes? And MASH1 transcription becomes from oscillatory to stable, while the P2X7 receptor would induce the transcription of neurogenin 2, and neurogenin 2 would become increased in its transcription expression rates, but it would not act anything on mesh 1. So, what it means, when mesh 1 induces neuronal differentiation and GABAergic differentiation, while when neurogenin 2 will block differentiation and maintain the cells at the neuroblast state. And this has some therapeutic applications, since we know that we have a loss of GABAergic neurons in Huntington disease. And now we will use this system to investigate Huntington disease. And this I'll show you here. Huntington disease is being caused by a mutation of a Huntington protein. And this Huntington protein has more polyglutamine residues as it should have. Normal are maybe 25, 30, but if it becomes more than 40, then with patients, they will develop Huntington disease. And this is a personality disorder, so it's also with uncontrolled movements, and it's clear that it has also effects on various brain areas, such as causing also depression, psychosis, and memory deficits, and finally, it will lead to death. So what I show you here is that Huntington's disease, here you see the expanded polyglutamine residues, you see this here, and if they're too much, then they cause the disease. Yes? But naturally, these regions here are important because they are thought to go into the nucleus and to control gene expression. And also, it's known that Huntingtin is a protein that is necessary for development. If you knock it out, when you see that you won't go behind the gastrulation state, okay? And this means, why is it so? There may be some hypothesis that we showed first. You see here, I will explain this later on. So this is a stem cell which expresses a normal Huntingtin protein, and here's one with 74 polyglutamines. And what you see, first of all, we express much more of Huntingtin than it should be. And the second is under these conditions that this disease-causing mutant is inducing in the gastrulation state the expression of brachyury. And this is actually a gene product which will activate mesodermal differentiation. So the hypothesis is here that this alteration changes neuroectodermal to mesodermal differentiation and will damage the nervous system. So now, how we can investigate this? So first of all, I was in the beginning talking to you about very shortly, without explaining what it is, about iPS cells. So we use induced pluripotent stem cells from the fibroblast of patients, human. We were reprogrammed, and then they are differentiated into neural progenitor cells. And the second model, what you're using, is here a CRISPR Cas9 modified mouse embryonic stem cells, where we knock out the endogenous Huntington protein from the mouse stem cells, and then we knock in a healthy variant of the protein or disease-causing variant of the protein. Okay, so then we can now study Huntington disease. And I only show you here very little experiments, but the most important one. So first of all, using the reprogrammed cells from the patient, there we can see that these cells, that they lose their spike activity. They don't have spike activity. You see, compared here to the wild type. And they have also, they don't have any, the peak height is also lost. And now, this is not all, but if we look now, to understand this experiment here we are measuring cell death on the left side using propylium iodide, which will go into the cell when the membrane is damaged, and the cell is going to die. So, and what you see here is control against tapsigagine. And tapsigagine, if you put it on a cell, what happens that it inhibits the reuptake of intracellular calcium into the endoplasmatic reticulum. So you have high calcium in the cytosol. And what we can see here, that basically in the absence and in the presence of tapsigagine, that we have an increase in cell death in the cell lines from Huntington disease patients. Yes, we see here the same on the right side. This is doing a similar experiment using labeling of apoptosis using annexin 5. Yes, and you can also see that these cells are the progenitor cells. You see here, they are more stimulated here by ATP, because then it shows you that there's a higher calcium peak. And this may also induce cell death, as high calcium concentration may be cytotoxic, okay? But now I come to the experiment which links Huntington disease to the P2Y2 receptor. On the upper panel, you see it's a neuroprogenitor stage, yes? And this is again the mouse stem cell line, which has been modified by CRISPR-Cas9, okay? Here you see the neurons. And you see, we've done many treatments with using ATP or the different concentrations, yes? But the most important thing is here, is using 2-SUTP, where you can see that you have a down regulation of P2Y2 receptor induced calcium transient in this Q74 cells. So it means they have the disease-causing mutation. Yes, you see also the same thing here. You see this in the MPC, and you see this in the differentiated neurons. And this means that the P2Y2 receptor is altered in its activity in Huntington disease using a stem cell model, okay? Yes, so I will now go very quickly to a second part to discuss this with you. Is an in vivo model where we studied a different disease. In this case, we studied the Parkinson's disease. And here, we are looking at specifically on the P2X7 receptor, because this receptor has been related with the induction of cell death and neuronal death. So very quickly, so neuroinflammation is a big deal in Parkinson's disease. During our life, we have a hormetic adaptation, and then we become old, we have inflammation. It means that we are not anymore only acute inflammation, but we have chronic inflammation which remain. And many of them may be caused also by a dysfunction in the gut. So that gut bacteria would even trigger immune cells to inflame the brain. But anyway, the inflammation from a peripheral side goes into the brain, and then it causes a redox dyshomeostasis and causes cell death. So now, So very quickly, what cells do we have in the brain which could do this? And here we have basically, we have microglial cells, yes, and we have also astrocytes, yes. So, and that means that we would basically recruit microglial cells and activate astrocytes, and these cells would respond to DAMPs. What is a DAMP? High ATP concentration is considered a DAMP, a danger-associated molecular pattern molecule, because high ATP concentration in the extracellular medium, they mean cell death. We have a release of pro-inflammatory cytokines, and we have also the activation of microglial cells. And then, because of this, and why the P2X7 receptor is linked to microglial activation, also to astrocyte activation, we say here, okay, it may be great to inhibit this receptor. And the first point, we want to inhibit the cytotoxic form of the receptor. And secondly, we want we want to inhibit the non-cytotoxic form of the receptor, which I mentioned during the beginning of my presentation. I say, okay, we need to downregulate these receptors to achieve neurogenesis. I mean, we could trigger neuroregeneration, yes? And this is a big deal, because for treatment, so L-DOPA treatment to re-putting dopamine into the brain is not very promising in the longer term. And also, if you transplant stem cells, they won't integrate. Yes, so the idea could be once that you deliver neurotrophic factors into the brain, or even a better idea, which to stimulate endogenous neurogenesis. And our hypothesis was to do this by blocking the P2X7 receptor. And what we see here, that is a work published 10 years ago, where Jonas Friesen showed you have neurogenesis in the striatum. So this may come basically from the subventricular zone, where the cells are attracted, and they may help to undergo regeneration processes. Yes, but now I will come quickly to the model. So for this, there are two types of Parkinson's disease. One of this disease is familiar, which is caused by mutations. And the second, much more frequent Parkinson's disease is believed to be toxin-induced during your life. You basically, but what happens is same what we do here is not more similar what you hear. So that were dopaminergic neurons in the substantia nigra and nigrostriatal pathway, and in the striatum will die. And we achieve this here in the mouse animal by injecting 6-hydroxydopamine only in one hemisphere. So we induce Parkinson's disease in one hemisphere. And what happens when? That these animals, they begin to rotate, because they cannot go any more straight, because one dopaminergic hemisphere is destroyed. Now, what we did very quickly, and that's what we showed here, number of rotation, which would is in line with the loss here of tyrosine hydroxylase staining in the injected hemisphere, because tyrosine hydroxylase stains dopaminergic neurons. Yes, and together with this, with rotation behavior, number of rotations per minute go up. Okay, yeah. And also you see that you have the same type of lesions here in the substantia nigra. So now again, why P2X7 receptor? We see, if we induce experimental Parkinson's disease, the receptor expression goes down to a massive loss of tissue and cell death. But then, finally, it comes back. It goes up, and this means it has functions in Parkinson's disease. And for inhibiting the P2X7 receptor, we use here brilliant blue G. This is a brain-variant P2X7 receptor inhibitor, and it's approved by the FDA, American FDA, okay, as a food additive, okay? So now, animals are injected 6-hydroxydopamine. After 1 week, we show that these animals are rotating. That means that one hemisphere of dopaminergic neurons is destroyed. Then we treat with BBG for 7 days, and then we check for behavior and for immunohistochemistry. And what you see here very quickly, so the idea how it shows really, so this is an untreated mouse which has, after 1 year of injection of 6-hydroxydopamine, yes, and the animal is going to rotate the whole time, okay? Now, next slide. We could show that this effect of 6-hydroxydopamine is actually dose dependent. So that we at 50 milligrams per kilo, we almost inhibit every rotation, and at 75 milligrams per kilo, there are no rotations after 1 week of treatment with the compound. And you see also here that with behavioral aspects, recuperation in motoric is in line with immunostaining for tyrosine hydroxylase positive dopaminergic neurons. You see, at a low concentration here, so of BBG, there is no regeneration, so there are no neurons. It starts with 50 milligrams per kilogram, and then it goes a maximum at 75 milligrams per kilogram, where you don't see a reasonable difference between the unlesioned control hemisphere and the lesioned hemisphere treated with BBG. Yes, and you see the same in the substantia nigra. You see also the recovery of dopaminergic neurons. And here we show basically the quantitative data of the recuperation of dopaminergic neurons using these types of conditions, okay? So now, I want to show you finally, I was mentioning it has to do with inflammation, yes? And if we treat the animals with BBG, we see also that we have much less IBA1 positive microglial staining. So it means that the inflammation process is being suppressed. So, and finally, I'm not talking too much, yes, I want just to go show you the final results here. But P2X7 receptor inhibition favors neuroregenerative and neuroprotective process in the red matter of Parkinson's disease, yes? And in the study before, using stem cells, now I showed you that P2Y2 and P2X7 receptors are antagonists in the progress of neural differentiation to GABAergic neurons. And that the P2Y2 receptor is dysfunctional in cells obtained from Huntington disease patients. It means, overall, that purinergic receptor modulation as a therapeutic strategy for the treatment of neurodegenerative diseases. Okay, now I would like here to thank my group. So it's done a lot of this work, but all the work, almost all the work on Huntington, Talita Klasa, and my doctoral student, Roberta Andres, with Agatha Oliveira Giacomelli, was a postdoc of mine. She's not anymore here, because she's not on the slide here. we are working on the Parkinson disease model. And I would also like to acknowledge my international collaborators like Ryoji Kageyama from Japan, Yang Diten from Harvard Medical School, and Yong Tang, Chengdu International Joint Research Center for Polynesia Signaling. I'm also making part of this center. And my local collaborators, Claudiana Lamio and Luis Brito from University of Sao Paulo. And finally, very finally, I would like to invite you for our meeting on Polynesia signaling, which will take place in Santos, Sao Paulo State, from November 5 to 8. And so the registration is open, and I think you can yet submit an abstract for a poster presentation if you would like. Thank you very much. I will close now the presentation, and I will get out of a presentation mode. And thank you very much, Rosa, for inviting me.
Thank you very much, Professor Henning, for your excellent lecture, and now the important results on the porinergic system that you showed us here. Thank you very much. E agora nós vamos seguir, então, com a sessão de encerramento do evento, né? Que foram esses 3 dias de apresentação. O evento foi chamado de Sinergia, nós falamos do primeiro dia, né? Ciência, Educação e Arte, porque para nós, né, durante todo esse tempo, é o sentimento que nós temos. 2018 iniciou o CAPES PrInt, nós estamos finalizando em 2025 agora, em fevereiro. Tivemos muitos avanços e durante esses 3 dias nós podemos também compartilhar um pouquinho daquilo que foi trabalhado. Nem tudo pode ser mostrado, né? E também o nosso primeiro evento, nossa primeira intenção era o evento presencial, que ocorreria em maio, e tivemos em maio, então, um evento climático no Rio Grande do Sul, e pensamos até em cancelar o evento, mas depois não, nós decidimos que ele aconteceria de forma online, como aconteceu agora. Então, muitos desafios durante todo esse nosso, digamos assim, pensar no evento e a execução dele. Também o professor de Virgílio faria a nossa palestra de abertura, né? Fizemos uma homenagem para ele no primeiro dia. Então, para nós também foi uma grande perda para a ciência, né, em nível internacional, e para nós, da sinalização polinérgica, né? Então, expressamos isso também no primeiro dia. E também eu gostaria aqui de deixar o meu agradecimento para todos os colaboradores durante, né, todos esses anos que fizeram o CAPES PrInt acontecer. Então, nós tivemos vários relatos aqui, todos nós compartilhamos a mesma intenção, que é a internacionalização da nossa universidade, né? Então, podemos dizer que nós tivemos avanços. Eu quero deixar aqui a minha gratidão, né, a ajuda de todos, novamente ao professor Henning, que fez a nossa, né, palestra de fechamento do evento. O professor Henning e eu trabalhamos juntos no Clube Brasileiro de Purinas, né? E ele é o presidente agora. Queres falar, Henning?
Yes, I will talk in English. First of all, I would like to dedicate my lecture to Professor Francisco Di Virgilio. He should be here, and it would be so important to have him here. And second, so I apologize that I did not respond to the collaborators of Nigeria, and I will respond, yes, because I was just seeing this. I was so extremely busy all the time with traveling that I did not write, but I will do so. Yes, so this I promise. Yes, so I will do this of this weekend and try to. Yes, yes. And of course, Horsa, you are absolutely. I think this is amazing. No? So your integration into the science of polynergic signaling, the excellence of your research, yes, and so how you're building up this big network for the university, Federal University of Santa Maria. And I think this is very important to integrate scientists from all over the world. That's what you do here. That's it. From Europe, you have from United States. You have collaborators who are from Nigeria, from Africa. And I think this is extremely important, but because in science, always we are interested in the same thing. So to promote science, and to promote knowledge. And if you promote basic science, you will also promote medicine and health, because you will understand what is happening and you will get better treatments. Yes? And in this way, I would just like to say that poietic signaling is very much going into this direction. Today, we recognize how important this is for all cellular processes, and on the cellular level and also on the level of the patient. So thank you very much, Rosa.
Thank you very much, Professor Henning. E agora passo a palavra ao Fernando.
Certo, obrigado, Rosa. Bom, então, chegamos ao fim do Sinergia, Ciência, Educação e Arte. Como disse a Rosa, ao longo desses 3 dias, podemos conhecer as pesquisas e os resultados apresentados dos relatórios, ainda parciais, das experiências proporcionadas pelo projeto CAPES PrInt, Estratégias Farmacológicas e Nutricionais para a Promoção da Saúde, que é coordenado pela professora Rosa, aqui na Universidade Federal de Santa Maria. Como dito também, a gente trabalhou com essa ideia da sinergia como um somatório de esforços individuais, gerando algo maior, impulsionado pelo vínculo entre todos os que participaram dessa jornada de colaboração. Então, que a ciência, a educação e a arte continuem se encontrando para criar novos horizontes. Muito obrigado a todos, agradecemos a todos aqueles que tornaram possível o evento, os parceiros institucionais do UFSM, conferencistas, palestrantes, ao público em geral e à equipe do Labart. Muito obrigado.
Eu também gostaria de agradecer à equipe do Labart pelo incansável empenho. Muito obrigado, minha eterna gratidão.
Nós estamos bem amparados, Rosa. Então, muito obrigado a cada um dos participantes pelo sucesso desse evento. Podemos encerrar a transmissão.
Okay, so thank you very much again. Yes, bye-bye.
Thank you, Professor.